Hello friends, you are watching Physiology Made Easy. Friends, we will try to understand Hemopoietic system in detail through this video series. If you like the video, please hit the like button. Kindly subscribe the channel and also press the bell icon to receive all notifications regarding more such informational videos. This is the fourth video in this video series and second part of video on red blood cells. In this video, we will consider sites of formation of RBCs, changes in RBCs during erythropoiesis, stages of erythropoiesis, regulation of erythropoiesis, factors necessary for erythropoiesis, and lastly, fate of RBCs. So, what is meant by erythropoiesis? Erythropoiesis means origin, development and maturation of RBCs. Let us consider the sites for production of RBCs. In fetal life, the erythropoiesis occurs in three stages: mesoblastic stage, hepatic stage and myeloid stage. In mesoblastic stage, during the first 2 months of intrauterine life, RBCs are produced from mesenchyme of yolk sac. In hepatic stage, from the 3rd month of fetal life onwards, liver is the main organ that produces RBCs. Spleen also produces RBCs during this stage. And in myeloid stage, during the last 3 months of fetal life, RBCs are produced by red bone marrow. After birth, up to the age of 20 years, RBCs are produced from red bone marrow of all bones. After the age of 20 years, RBCs are produced from the membranous bones like vertebrae, sternum, ribs, scapula, iliac bones and skull bones, and from the ends of long bones. In erythropoiesis, following changes occur in RBCs. There is reduction in the size of the cell. Next, there is appearance of hemoglobin. At the intermediate normoblast stage, hemoglobin synthesis starts. Next is disappearance of nucleus. During the late normoblast stage, nucleus disappears from the cell. During maturation of RBCs, other cell organelles such as ribosomes, endoplasmic reticulum, Golgi apparatus, mitochondria, they also disappear. And lastly, change in cytoplasmic staining. Before the appearance of hemoglobin, cytoplasm is basophilic. When hemoglobin synthesis is almost completed, cytoplasm is stained by acidic dyes. Next are the different stages of erythropoiesis. The primary cells which give rise to blood cells are called stem cells or PHSC or pluripotent hemopoietic stem cells. In early stages, these cells are not designed to form a particular type of blood cell, hence they are called uncommitted PHSC. When the cells are designed to form a particular type of blood cell, they are termed as committed PHSC. Next are the cells from colony forming unit erythrocytes. The cells from these units are derived from stem cells. Next is proerythroblast. It is the first cell derived from colony forming unit erythrocytes. Diameter is about 20 microns. Its nucleus is large and it occupies cell almost completely. There are about 2 to 3 nucleoli in nucleus. The cytoplasm is basophilic in nature. It undergoes multiple divisions to form early normoblast. So early normoblast, it has a diameter of about 15 microns. In the nucleus, nucleoli disappear. Condensation of chromatin network occurs. Cytoplasm remains basophilic in nature. Next stage is intermediate normoblast. Diameter of the cell is about 10 to 12 microns. Nucleus is still present, but the chromatin network shows further condensation. The hemoglobin starts appearing in the cell. In this stage, cell stains with basic as well as acidic dyes. Next stage is late normoblast. Diameter of the cell further decreases to about 8 to 10 microns. Nucleus becomes very small with very much condensed chromatin network and it is termed as ink spot nucleus. Quantity of hemoglobin increases. Cytoplasm becomes almost acidophilic. In the final stage of late normoblast, nucleus disintegrates and disappears. After the loss of nucleus, the normoblast passes into the next stage, that is reticulocyte. It is slightly larger than mature RBC. The cytoplasm contains reticular network due to which the cell is termed as reticulocyte. In adults, reticulocyte count is about 1%. Reticulocyte matures to form erythrocyte. The reticular network disappears and the cell becomes mature. It attains biconcave shape. The cell decreases in size to about 7.2 microns in diameter. So it takes about 7 days for the development and maturation of RBCs from proerythroblast to mature erythrocyte stage. It requires 5 days up to the stage of reticulocyte, and reticulocytes take 2 more days to form mature RBCs. Next is the erythropoiesis regulation. The most important general factor necessary for erythropoiesis is the hormone called erythropoietin. It is also termed as hemopoietin or erythrocyte stimulating factor. Let's see its action in detail. Hypoxia is the stimulus for secretion of erythropoietin. Hypoxia may be due to any reason such as less number of RBCs in blood, as in anemia, poor blood flow, or pulmonary diseases. In response to hypoxia, peritubular capillaries of the kidneys secrete major quantities of erythropoietin. Erythropoietin acts on stem cells present in the bone marrow and causes their differentiation. It also promotes every stage of maturation of RBCs as well as their release into the peripheral circulation from bone marrow. Thus, it increases the number of RBCs in peripheral circulation, which now carry more amount of oxygen, and thus hypoxia gets corrected. About 90% of the erythropoietin is formed in kidneys and the remainder is formed mainly in the liver. So this mechanism, it is the most powerful and most important mechanism controlling the RBC production. Let's now see the factors needed for erythropoiesis. General factors needed for erythropoiesis include: first one is erythropoietin that we have discussed right now. Hormones like thyroxine and androgens, both these hormones enhance the erythropoietin secretion. Hence, they also increase the production of RBCs. Vitamins such as vitamin C, vitamin D, vitamin E are also necessary for erythropoiesis. Next are maturation factors. These are vitamin B12, intrinsic factor of Castle, and folic acid. Intrinsic factor of Castle is produced in gastric mucosa by parietal cells. It is required for the absorption of vitamin B12 from intestine. In the absence of these factors or vitamins, RBCs do not mature and remain larger in size. So it leads to megaloblastic anemias. Lastly, for the synthesis of hemoglobin in RBCs, some factors are necessary. These are first class proteins which are required for the formation of globin part of HB. Iron is necessary for the formation of heme part of hemoglobin. Some minerals like copper, cobalt, nickel, and vitamins such as vitamin C, nicotinic acid, pyridoxine, etc. also are needed for hemoglobin synthesis. Next point is fate of RBCs. After their formation in red bone marrow, RBCs are released into peripheral circulation. Normal lifespan of RBCs is about 120 days. When the RBCs become old, their wall becomes weak, they lose their flexibility, and the shape is also changed. Most of these RBCs are broken down while passing through spleen, as lumen of splenic capillaries is much less. These broken fragments as well as worn out RBCs are phagocytosed by the reticuloendothelial cells or macrophages present in spleen. Macrophages present in liver as well as bone marrow also phagocytose senile RBCs. In macrophages, hemoglobin from the fragmented RBCs is split into heme and globin. Globin is converted back into amino acids, which are further reused for protein synthesis. Macrophages release iron from the heme part of the hemoglobin and pass it back into the blood to be carried by the transferrin either to the liver and other tissues for storage in the form of ferritin or to the bone marrow for the production of new RBCs. The porphyrin portion of the heme is converted by the macrophages into the biliverdin, which is then converted to bilirubin. Bilirubin is released into the blood and later secreted through the liver into the bile for its removal. This is all about red blood cells. If you haven't watched the previous videos in Hemopoietic System series, links are provided in the description below. Upcoming video will be regarding hemoglobin and anemias. Thank you very much.